Welcome back to the SmartSoleHub Podcast. I'm Jae Kim. Today we are tackling the elephant in the room. It is the single biggest clinical paradox in all of perineural injection therapy, and it is a question almost every sharp patient will eventually ask you. If chronic high blood sugar destroys peripheral nerves and causes diabetic neuropathy, why on earth are we injecting pure sugar water directly onto nerves to heal them? How can glucose be both the poison that destroys the nerve and the medicine that repairs it? For a long time, we didn't have a perfectly packaged answer for this. We knew clinically that our 5% dextrose injections worked. We knew they were incredibly safe, but explaining the precise biological difference between diabetic nerve damage and dextrose nerve healing was tricky. Enter the Chang review. Published recently in 2026, Chang and colleagues put together a comprehensive paper looking at glucopuncture, which is the emerging umbrella term for injecting 5% dextrose for pain management. They synthesized the existing literature to explain exactly why this paradox exists, breaking the healing effects of dextrose down into 4 distinct mechanisms. But before we look at those 4 mechanisms, I want to use a simple metaphor to anchor how we think about glucose exposure. I want you to think about watering a house plant. If you put a plant under a gentle sprinkler for 10 minutes, it drinks the water, metabolizes it, and thrives. That is an acute beneficial pulse. But if you completely submerge that exact same plant in a bucket of water for a month, the roots rot and the plant dies. That is chronic toxicity. The fundamental difference isn't the water itself. The water is identical. The difference is the duration of the exposure and the ability of the system to drain. Keep that in mind as we walk through the 4 mechanisms outlined in the Chang review. The first mechanism is metabolic rescue. When a peripheral nerve is compressed or injured, its microcirculation is compromised. The nerve enters an energy crisis. It doesn't have enough ATP, the cellular energy currency, to run its sodium-potassium pumps. When those pumps fail, the nerve depolarizes. It starts firing off erratic ectopic pain signals. When we inject a bolus of 5% dextrose, we are delivering an immediate acute pulse of metabolic fuel. The starved nerve acts like the plant under the sprinkler. It rapidly takes up the glucose, undergoes glycolysis, produces ATP, and restarts those failing pumps. The erratic pain signals stop because the nerve finally has the energy to stabilize itself. The second mechanism is receptor resetting, specifically looking at the TRPV1 receptor. TRPV1 is an ion channel on the nerve that detects heat and burning pain. It is heavily involved in neurogenic inflammation. In a state of chronic diabetes, where the nerve is drowning in the bucket of water, the constant, relentless exposure to high glucose permanently damages and upregulates this receptor, leaving the patient in constant burning pain. But the Chang review highlights that an acute application of extracellular dextrose actually does the exact opposite. A targeted short-term pulse of dextrose binds to the nerve and downregulates the TRPV1 receptor. It temporarily closes the calcium channels and resets the nerve's pain threshold. The third mechanism is cellular repair, driven by the Schwann cells. We actually covered this extensively in part 2 when we looked at the Zhu paper, but the Chang review reinforces it beautifully. A temporary acute spike in local glucose tells the Schwann cells to release nerve growth factor and BDNF. It kicks off a regenerative cycle. But in unmanaged diabetes, the glucose never leaves. The signal never turns off. The Schwann cells become exhausted, eventually stop producing growth factors, and undergo destructive changes. Again, duration makes all the difference. The fourth and final mechanism is simply the dilution factor. When a patient has poorly controlled diabetes, their entire systemic environment, every capillary, every tissue bed, is saturated with elevated glucose 24 hours a day, 7 days a week. When we perform perineural injection therapy, we are injecting a few milliliters of 5% dextrose locally along a Valix point. It doesn't just sit there forever. The body immediately goes to work, the interstitial fluid buffers it, the surrounding tissues metabolize it, the vascular system clears it, the nerve experiences a temporary, highly beneficial metabolic spike, gets the energy and the repair signals it needs, and then the local environment returns to normal. So, what does this actually mean for us in the clinic? First, it gives us the clinical confidence to look a diabetic patient in the eye and explain exactly why our dextrose injection will not make their neuropathy worse. We can explain that we are providing an acute energy rescue, not contributing to a chronic toxic bog. Second, it completely validates the biological mechanisms we've been discussing over the last 3 episodes. It ties the metabolic rescue, the TRPV1 downregulation, and the Schwann cell activation into one cohesive, evidence-based model. And finally, it reinforces why our treatment spacing is so critical. We want to deliver the metabolic pulse, trigger the healing cascade, and then get out of the way. We must allow the local environment to clear and the repair cycle to run its course before we introduce another stimulus. We want to be the sprinkler, not the bucket. You can find my full clinical breakdown of the Chang review, along with a summary of the 4 mechanisms, over at jaekim.com.au/blog. Thanks for listening. Next time, in part 5, we are going to dive into a 2003 paper by Tan and colleagues that explores the cellular break, and we'll ask the final lingering question: how often is too often when it comes to injecting nerves? I'll see you then.